Hello everyone, today we're going to talk about microbial growth. Bacteria have different requirements for their growth. Um, Not all the bacteria are going to need the same elements to grow. Not all of them are going to prefer room temperature. So again, each bacteria is different. So when we come and think of what are some requirements that are pretty important to have optimal bacterial growth, and so there are physical requirements such as the temperature, the pH, the osmotic pressure, and there are also chemical requirements. So what type of elements do these bacteria need, again, for them to grow and thrive? Do they need nitrogen or phosphorus? Do they need some like a source of carbon or a source of sulfur? Vitamins, amino acids. So all of these things are pretty important to talk about when we are discussing bacterial growth. Even oxygen. So the requirement of oxygen, how much oxygen do these bacteria need? And of course, not all the bacteria are going to have the same requirements. So depending on the bacteria that we're talking about, for example, some bacteria are obligate aerobes, which means that they do require oxygen and they rely on the oxygen. So they need high concentrations of it. If we talk about obligate anaerobes, those bacteria they do not need any oxygen, and actually if the environment has oxygen, then it is pretty toxic for them, so they will tend to survive in environments that have basically no oxygen. And then we have some bacteria that are facultative anaerobes, and so those bacteria, for example, they can tolerate, they can basically survive in either aerobic conditions or anaerobic conditions, but they would tend to prefer aerobic conditions. Um, so as you can see, each each type of bacteria can have different requirements, and that's not only, that's not just for the oxygen. For the temperature, some bacteria prefer warm temperatures, some prefer really cold temperatures. Most of the bacteria, though, they prefer room temperature. So we can, if we are trying to grow bacteria in the lab and you just leave your agar plate at room temperature, you are going to have lots of bacterial growth because, again, lots of bacteria, they do prefer room temperature. pH, most bacteria, they prefer neutral pHs. Very few bacteria prefer acidic environments. So again, when we go ahead and we prep the media to grow our bacteria, most of the time we just prep the media so that, again, it has neutral pHs so that we can grow a bunch of different types of bacteria, unless we know that the bacteria that we are trying to grow prefers acidic environments. Then we go ahead and we prep the agar, prep the media, basically make it acidic. So we're going to talk about each one of those in a bit more detail. If we look at this graph right here, where we are graphing the temperature and the rate of growth of the bacteria, as I was saying, most microorganisms, they do grow at room temperatures and at temperatures that basically humans favor. But of course, we do have differences between the microbes out there. So some of them prefer much cooler temperatures or room temperature or much warmer temperatures. So if we go ahead and take a look at a few examples, we do have bacteria that are known as psychrophiles, and as you can see, psychrophiles, they can grow at cooler temperatures. Their optimal growth, which is right here, it's around, I would say, like 11 or 12 degrees Celsius. The psychotrophs, they prefer much warmer temperatures compared to the psychrophiles, and their optimal temperature up here would be around 23 or 24 degrees. So kind of around room temperature. That's where actually they would grow and thrive the most, around room temperature. Mesophiles, as you can see, they prefer even warmer temperatures, and again, they would grow optimally around 35 degrees Celsius. Then we move on to the group that are preferring even warmer temperatures, and those are the thermophiles, and you can see that their optimal growth would be around 62 or 65 degrees Celsius. And then we have the hyper thermophiles, and those are the ones that prefer much warmer temperatures. And as you can see, they do really great when the temperature is pretty high. So those would be the hyperthermophiles. So as you can see, different microorganisms would have different requirements when it comes to the temperature. And as you can see right here, even though each group, they can grow at a range. So for example, like thermophiles, you can see that they can grow anywhere from 40 degrees Celsius to 70 degrees Celsius. Mesophiles, for example, from 10 to 50. So of course, there's a wide range, but each of these microorganisms would have an optimal, an optimal growth. And so right here you can see the peak of the curves would represent those optimal growths. This image is just showing you again the temperatures, Celsius or in Fahrenheit, and it's showing you where the bacteria would tend to grow rapidly, or where they would grow very slow. So you can see right here that rapid growth of bacteria happens right here within this zone. So anywhere from like 12 degrees Celsius to around 50 degrees Celsius, or basically from around 60 to 120 Fahrenheit. And that's why we call it the danger zone, because this is where most of the bacteria, they grow pretty rapidly, and they can start definitely producing lots of toxins. As the temperature rises, and then you usually have slower bacterial growth, because higher temperatures, most bacteria cannot really tolerate such high temperatures except a few. But that's why when we want to get rid of most of the bacteria that we grow in the labs, we go ahead and we autoclave it. And so when you place it basically in the autoclave, you set the temperature to a very high temperature, and you also add moisture, and usually heat and moisture kills the bacteria. You can see that as the temperature also cools down, then you're not going to have significant growth, especially below freezing. And that's why the most common way, for example, for us to preserve our food, we go ahead and we refrigerate it, because usually refrigerator temperatures do slow down the growth and spoilage caused by the bacteria. And again, very few pathogens would be able to tolerate refrigerator temperatures. If you recall, we said that bacteria grow rapidly at temperatures that are like room temperature or a bit higher, so anywhere from like 20 Celsius to around 40 Celsius or so. So right here you can kind of see that this would be the danger zone. This is where you would have a lot of the bacterial growth. And so for us to preserve our food and basically repress that bacterial growth, we go ahead and we place our food in the fridge. Now, what this graph is trying to show you, though, is that if you have two pans and they are, let's say they contain rice, but this one right here, it's not as thick, like the pan or the width of it, as this one right here. So this one definitely has more food in it. It has more depth to it. You will notice that this one right here, it's actually going to take way longer before it cools down. So notice how it's taking many hours, and you can see that it's going to spend quite a few hours in that danger zone, and that could mean that the bacteria could definitely start growing. Even though you've placed it in the fridge, it's not going to cool down as fast compared to this pan right here that doesn't have as much food, because depth-wise it's less. And you can see that only within like an hour and a half or two, you can see that it's going to, temperature wise, it dropped dramatically and you're not anymore in that danger zone. And then you can see that it's going to cool down even more rapidly compared to the pan that has more food in it. Another important factor that we need to talk about when it comes to bacterial growth is the pH of the environment. So most bacteria, they grow at neutral pHs, or pHs that are pretty close to being neutral. So there's a range usually from 6.5 to around 7.5. This is where the bacteria would love to grow. This would be their optimal pH level. And again, of course, depending on which bacteria you're talking about, the range is going to vary from one bacteria to another. Molds and yeast, fungi, they tend to grow at much acidic levels, so at pHs of 5 and 6. And that's why when we grow bacteria in the lab and we prep our media, we go ahead and set the pH of the media to the ranges that the bacteria would prefer. But in the lab, if we want to go ahead and grow yeast or basically fungus, what we do is we usually use the Sabdex plates, because we usually set the pH for the Sabdex plates to around 6 or 5. So that would enhance the growth of the fungi and make it harder actually for the bacteria to grow on the Sabdex plates. But there are some bacteria actually that do prefer much acidic environments, and we refer to them as acidophiles. Another thing to consider when we are talking about bacterial growth is the osmotic pressure. So when we are talking about osmotic pressure, we need to think about the environment that those bacteria would be living in and how much solutes would this environment have. Now, for the bacteria for most of them to grow and thrive, you don't want to have too many solutes outside the bacteria, because if the condition outside is basically hypertonic, which means it has a high concentration of solutes, what's going to happen is the water is going to leave the bacteria cell, because water likes to go from a low solute concentration to a high solute concentration. And so if the condition outside is basically pretty high, in terms of the concentration of its solutes, then the water is going to start exiting out of the bacteria. And why is that bad? Because when lots of water exit the bacteria is going to start to shrivel up, and it can die. And you can see right here that lots of water is exiting out and the plasma membrane is collapsing, and we refer to that as plasmolysis. And again, that was due to the high osmotic pressure, and the water was rushing out of the bacteria at a high rate from the low solute concentration inside of the bacteria to the high solute concentration outside of the bacteria. Actually, some bacteria, they are either known as extreme, or we can refer to them as obligate halophiles, and that means that these bacteria, they actually thrive when the solute concentration is actually pretty high. So they actually require a high salty condition, or you can say that they require a high osmotic pressure. Facultative halophiles, those are the ones that can tolerate high osmotic pressure, but they don't require it. But they can definitely tolerate it, and they are able to survive if the condition outside of the bacteria is highly salty. And that's why to preserve our food, we usually add lots of salt. For example, you would get a piece of fish, and to preserve it, you go ahead and like put lots of salt on it, or you go ahead and place the food in a high, doesn't have to be high salty condition. It could be like a high sugary condition. So you could preserve things in honey, or you can, for example, like if you think of the condensed milk, it's extremely sugary. And so high salt or sugar concentration, what would it do? It would draw the water out of the microbe cells, and therefore it would prevent the growth of the bacteria. So these are some ways that we could, use to preserve our food. Basically, place the food in a very high salty condition or a high sugary condition to basically prevent the bacteria from growing and causing food spoilage. When we talk about bacteria growing in nature, bacteria in nature, they tend to live in communities, and we call these communities biofilms. These biofilms, they tend to adhere to surfaces, whether these surfaces are plastic surfaces or rocks or even your teeth. So the bacteria growing on your teeth, they tend to clump together, and you would have that biofilm on your teeth, and they can even survive in water. So these biofilms, these communities of bacteria, can definitely survive underwater, even if it's a river and basically like streams, so the water is always moving with definitely high pressure. They can still actually survive. And so biofilms definitely create a problem, and they can cause so many health issues, because if you have a biofilm of bacteria growing on medical utensils, or for example on within catheters, that's going to cause problems, and it can cause diseases to the patients. And so biofilms, it's really hard to get rid of because the bacteria, as I was saying, they live in communities, and even if they go ahead and they migrate, see how right here, like a whole clump of them, they move together, they migrate together. And so when all of these bacteria are basically living together, what they start doing is they start sharing their nutrients, they start transferring some genetic information amongst themselves. And what happens is if one of those bacteria living in this community is carrying a gene that makes it resistant to certain antibiotics, or it's carrying a gene that would make it resistant to phagocytosis, it can actually transfer over those genes to another bacteria. And so you would have another bacteria that wasn't resistant to antibiotics. Now it is resistant to antibiotics. And so that's why lots of these biofilms are definitely harmful, and they can, it's really hard to, as I was saying, to get rid of them, as they could be resistant to your attacks from your immune system. They are resistant to antibiotics, etc. And when we talk about different groups of bacteria, not all bacteria are harmful, not all of them are pathogenic. And so when we talk about the biosafety levels, biosafety level number 1 and 2, usually that's in the labs. So we deal with bacteria, usually environmental bacteria or bacteria that are not pathogenic, and so therefore we still need to put on our lab coats and our gloves and eye protection like our goggles. You basically have to put on your PPE. But when we are talking about bacteria that are definitely harmful and pathogenic and pretty dangerous, then that would be a level BSL-4, and that's when the scientists working in these labs, they would have to put on suits like the ones that you are seeing right here in the picture. And not all labs out there are capable of working with such pathogenic, harmful microorganisms. So for example, when we think of microorganisms that are pretty dangerous, like the coronavirus or the Ebola virus, for example, because those viruses are super dangerous, they can only be handled in a BSL-4 lab. And as I was saying, we only have a handful of these labs in the United States. Those labs, they tend to have an atmosphere that's under negative pressure, so that aerosols containing pathogens would not escape. And definitely the scientists working there, they have to put on these suits right there that you're seeing, and those suits, they are usually connected to air supply, and the air pressure inside of the suit would have to be higher than the atmospheric pressure, and that's so it can basically prevent any microbes from entering into their suits. To preserve bacteria, so let's say you grew some bacteria and you want to go ahead and preserve it, and you want to preserve it for a while, then what we could do is something known as deep freezing, and that's where you go ahead and you basically freeze your microbial organisms, -50 degrees to -95, and then if you need them, you can go ahead, thaw them, and then culture them And so this is one way for us to preserve the bacteria for several years. Of course, if it's just for a few days that you want to preserve your bacteria, you wouldn't go through the trouble of deep freezing. You would just take your plate and refrigerate it. But if you want to preserve it for several years, then you can consider this option right here, the deep freezing option, or you can consider the option of freeze drying, and that's where you go ahead and you freeze your bacteria sample, and then you go ahead and dry it, and you get that powder-like residue, and so then you take that remaining so you take that powder-like residue that you now have, which contains the bacteria, and then you can actually store it for years, and then when you want to revive it, you can actually revive it by hydrating it. So that's known as the freeze drying method. When we think of how bacteria divide, we think of two processes, the binary fission and the budding process. So this is the budding process right here, where you have the bacteria and then a bud starts to form, and the DNA, after it has duplicated, it starts, that duplicated DNA starts to move into the new bud, and then this bud detaches from the parent bacteria cell, and the bud that forms doesn't actually have to be the same size of the parent bacteria cell. The other process is binary fission, and you can see right here, here is the bacteria with its cell wall and plasma membrane, and it starts to duplicate its own DNA, and then the cell wall and the plasma membrane, they start to actually constrict, and you can see that kind of at the two poles you will have the DNA, the old DNA and the replicated DNA right here, and the cell wall starts to, like a new cell wall starts to form, and the plasma membrane, and those two cells, they completely separate, and you end up with two cells that are identical to one another and also identical to the parent cell. Now, when we think of the generation time, like the time required for the cell to divide, it can start dividing anywhere from 20 minutes after you went ahead and you've inoculated, let's say, on a plate. So if you wait for about 20 minutes, you are actually going to start seeing new bacterial growth, or it can take a bit longer. So anywhere from 20 minutes to 24 hours, that's the time that's going to be needed for the bacteria to start to grow and multiply. And when we talk about the binary fission, as I said, the cell ends up like splitting itself into two. So when we come and try to calculate the number of cells that we can have, we just say 2 to the power of a number of generations. So if the bacteria has gone through 2 generations, you would say 2 to the power of 2, so you would get like 4 cells. If the bacteria has gone through like 5 generations, then it would be 2 to the power of 5. And so if you take a look right here at this table, you can see so that if the bacteria has gone through 5 generations, how many cells can I have? I would just say 2 to the power of 5, so 32. If my bacteria has gone through 10 generations, then I would say 2 to the power of 10. That means I have a bit more than 1,000 cells growing, etc. And you can see right here that the growth is going to be exponential. So the bacteria definitely grows exponentially, and it grows really fast. when you give it the correct environment, like you place it at the correct temperature, pH, you provide it with the nutrients and the vitamins and proteins that it needs, then the bacteria is going to grow, and as you can see, it does grow exponentially. There are four bacterial growth phases: the lag, log, stationary, and death. So let's say that you're growing a bacteria on a plate in the lab. What are the four stages? So the first stage would be the lag phase. And the lag phase, if you take a look at it, you notice that you don't have any increase in the number of bacterial cells, which tells you that the bacteria is not duplicating or multiplying itself yet. And the reason why you don't see any growth right there is for the lag phase, or during the lag phase, what happens is the cell is just prepping itself for the vision. It's making the enzymes that it needs, etc. And the thing is also that the bacteria, when you've went ahead and you've swabbed it onto a plate, you're basically placing it in a new environment that's not used to yet. And so therefore, during the lag phase, you're not going to see any bacteria cells increasing in number. During the log phase, you will see exponential growth, and the bacteria is definitely multiplying and dividing itself. So right here, the log phase, you do see the exponential growth. And then there's the stationary phase, and you can see that the growth rate plateaus out, and that's because the number of deaths are actually balancing the number of new cells that are being made, and the amount of nutrients on the plate is starting to decrease, the amount of water is starting to decrease, even the space on the plate is also decreasing, so that's why you get this stationary phase right there. And then you can see the graph is dipping downwards, and that's because this is known as the death phase, and that's when the number of deaths eventually exceed the number of newly formed cells. You see that drop right there. There are several different methods to actually count and figure out our microbial growth. So three methods that we're going to talk about: plate count, filtration, and direct microscopy count. When we go ahead and we plate and we inoculate our plates with the bacteria, we usually dilute first our bacteria. So we would usually grow our bacteria in a liquid nutrient broth, and then we go ahead and we dip our cotton swabs in the liquid broth, and then we go ahead and we start inoculating our plates. And we usually go through serial dilution. So we usually take that broth that's now containing bacteria, and we go ahead and we dilute it out. And then after we dilute it out, we go ahead and we take a sample of each and we grow it on our plates, and we are after we plate it, we want to use the plate that has anywhere from 30 to 300 colonies growing on it, and we refer to them as CFUs, colony forming units. Now, we usually, as we are spreading the bacteria on our plate, as I said, we usually either spread it with the cotton swab, or we usually use a spread plate that would go ahead and spread the bacteria on the plate. As you can see right here, you start off with the original inoculum or your original concentration, and you can see right here we are doing tenfold dilutions. And so you would basically, for example, take a 1 milliliter out of your original concentration, place it in a test tube, and then add 9 milliliters of the water or the broth. And so this would be a 1 to 10 dilution. If you continue doing that, taking 1 milliliter out of the sample and then placing it in a 9 milliliter fluid, that would be another tenfold dilution. And after you go ahead and you get your test tubes ready, you go ahead and you start plating it, and you can either take 1 milliliter to plate it, you can take less, you can take more from that test tube, and then after you get your plates, you go ahead and you figure out which one which plate is going to be used. So which plate has a good amount of colony forming units, and so you want to look for the one that has anywhere from 30 to 300 colonies, nothing less than 30, nothing more than 300. So it will probably be this plate right here if we count the colony forming units. So notice how this one only has 3 colony forming units, so I wouldn't choose this one. And then these right here are too many to count, so those are not going to be used. So right here, this one, whatever number of colonies, if it is between 30 to 300 colonies, then that's the plate that we're going to go ahead and use and do further testing on the bacteria that's growing on that plate. When we pour our plates, there are 2 methods. So either you have the agar already set and you just pour whatever amount of the broth onto your plate, and then you go ahead and you spread it. Usually you have that spreader, kind of like a hockey stick that you use to spread the bacteria throughout the whole entire plate. And then you go ahead and you check out the colony forming units that you get. Another way is to mix in the agar with the bacteria sample. And when you do that, when you mix the two together, you're going to have some colonies that are growing on the surface of the plate, and then you're going to have colonies that are within the agar, like deep within the agar. This is usually called the pour plate method, and this side right here is usually known as the spread plate method. Another thing is the process of filtration. So for filtration, you usually get like a sample, let's say from a lake or from a stream or basically any source of like water. And then we go ahead, take that water, we pass it through a very thin membrane filter that has very small sized pores, and those pores are extremely small to the point that it's not going to allow the bacteria to pass through it. And then you take that filter membrane and then you transfer it over to a nutrient agar. One way to figure out how much bacteria is growing, you go ahead and you take a sample of, let's say, the broth that you have bacteria growing in it, and you place it in kind of like on a slide that has grids, and it has like large squares, and then after you place your bacteria onto that grid, you go ahead and you place a cover slip, and then you go ahead and you read it. You can actually put it and scan it in a machine, and what it does, the machine is going to tell you how many bacteria cells you have there. And the way they do it is they look at one large square and then they count how many bacteria is within that large square. And again, usually this is computerized, and the computer would be able to read it pretty quickly, and you would get the results also pretty quickly. Let's say that you've swabbed a surface and you went ahead and you've added that sample into a test tube with broth, and you've incubated it for several days so that the bacteria can start growing within the broth solution. And now you want to figure out if you have a high amount or a high concentration of bacteria within that broth. An easy way of determining whether you have a high concentration or a low concentration of bacteria within your broth, that you would take your test tube, like your broth test tube that has been inoculated with a bacteria, and you place it in a spectrophotometer machine. The spectrophotometer machine is going to be able to give you data about how much light is being reflected off. So the more light that shines through and gets reflected off of your test tube, that means that it has a pretty low concentration of bacteria. If your test tube has lots and lots of bacteria growing in it, when you go ahead and shine the line through that test tube, a very minimal amount of light is going to be reflected. And that's because most of the light is being absorbed, and that's because you have a high concentration of bacteria within that test tube right there.